Good morning. Over the next 2 hours or so, we are going to discuss what happens to the different food items that we ingest.
As you know, the food that we ingest can be in the form of carbohydrates present in bread, rice, pasta, etc., or in the form of proteins found in meat, steak, chicken, fish, or in the form of fat which is present in butter, oil, etc. And the problem with these food items is that they are formed of macromolecules, so these are large molecules that cannot be absorbed by our gastrointestinal system, so they have to be broken down into smaller molecules so that they can be absorbed. And formation from large molecules to smaller molecules is called digestion, and this occurs in the presence of hydrochloric acid in the stomach, in the presence of enzymes secreted by the pancreas and salivary glands, and also in the presence of bile secreted by the liver. And this secretory behavior of the intestine is called secretion. After these large molecules are transformed into smaller molecules, they are absorbed by our small intestine, and this is what we call absorption. And during this digestion and absorption, the food particles present in the intestine are mixed with the pancreatic enzymes and bile, and then they are brought close to the mucosa so that they can be absorbed, and in the meantime they can be transformed or moved from one part of the intestine to the other, and this is what we call motility or motor behavior. And when we talk about a response to a meal, we have always to think about these four processes: digestion, absorption, secretion, and motility. So this is what we are going to discuss. First, we're going to talk about the general anatomy of the gastrointestinal system, the gross function of each organ. Then we'll talk about digestion and absorption of nutrients, carbohydrates, proteins, fats, and we'll talk about vitamins absorption. And then we'll talk about what regulate these GI processes. We will talk about neural and hormonal regulation, and what are the phases of these regulations, like cephalic, gastric, or intestinal. And then we will follow a meal from the time of chewing to the time of defecation. So the gastrointestinal system is formed by a long hollow tube extending from the mouth to the anus, and also by accessory organs which do not form part of the tube, but they secrete their contents through ducts into the tube itself. And these are the salivary glands, the liver, the pancreas, and the gallbladder.
The first part of the GI tube is the esophagus. It's just a tube that acts as a conduit to get food from the mouth and pharynx to the stomach. It has two sphincters, the upper esophageal sphincter and the lower esophageal sphincter. The lower esophageal sphincter plays an important role in preventing acid in the stomach from going up the esophagus. When the lower esophageal sphincter fails in doing this job, gastroesophageal reflux disease, what is called GERD, may occur.
Now we move distally to the stomach, which can be divided anatomically into three regions: the upper part, which is the fundus, then we have the body of the stomach, and the last part is the antrum, which has thick wall, and it's important for grinding of food. Now the stomach secretes hydrochloric acid, and also secretes some enzymes like pepsinogen, which is important in the digestion of protein. And also there are some scattered cells in the stomach that secrete hormones like somatostatin, and also other cells that secrete histamine. The stomach ends by the pyloric sphincter, which is important sphincter that control the passage of food from the stomach into the first part of the small intestine, which is the duodenum. Anatomically, there are multiple invaginations in the wall of the stomach, forming what we call gastric pits or gastric glands. And in these glands we find the cells that secrete acid, like parietal cells that secrete acid and intrinsic factor, and we find chief cells that secrete pepsinogen. And also there are important cells that secrete a mucus, and these play an important role, as we'll see later on, in the protection of the stomach from the acid secreted by parietal cells. As we said, the stomach secretes large amount of acid, and it's approximately 2 liters of hydrochloric acid every day, with a concentration of 150 millimolar, which is 3 million times more than the concentration in blood. And this occurs secondary to the presence of a proton pump, which is called hydrogen potassium ATPase, which is present on the luminal membrane of the parietal cell. This pump secretes H+ outside the cell into the lumen, and potassium is absorbed. Then potassium will leave the cell through a different channel. Now, this hydrochloric acid comes from the reaction between CO2 produced by the cell and water in the presence of carbonic anhydrase, with the formation of H+. At the same time that we have secretion into the HCl into the lumen, we have secretion of bicarbonate into the blood. So if you think about it, if we have a person who continues to vomit, for example, this person will lose H+, and will lose potassium. So he will develop alkalosis and decrease in potassium, and this is what we call hypokalemic alkalosis. How does the parietal cell secrete acid? Now, acid is present in the parietal cells inside vesicles, so that it can protect the parietal cells from the function of HCl. Now, the parietal cell has 4 important receptors: receptor for gastrin, histamine, acetylcholine, and somatostatin. The first 3 will increase secretion of HCl, whereas somatostatin inhibit secretion of HCl from the parietal cells. So when these receptors are stimulated, the vesicle will go and it will join it will fuse with the apical side of the parietal cell and start secreting H+ into the lumen. In addition, as we said, the stomach secretes pepsin, and pepsin is secreted in inactive form called pepsinogen, which is secreted by chief cells, and the secretion is usually regulated by acetylcholine. So this inactive pepsinogen, in the presence of H+, will be transformed into the active pepsin, and pepsin further transforms pepsinogen into pepsin. And this is important, as we will see later on, in the digestion of proteins. And it's important to know that pepsin is active at acid pH, so acid between 1.8 to 3.5, and if the acid of the pH in the stomach goes up, the pepsin will be inactivated. Both HCl and pepsin are important for protein digestion.
The question now is, how does the stomach protect itself in the presence of hydrochloric acid and pepsin? Well, as we have mentioned previously, there are cells in the gastric pits that secrete mucus, and mucus plays a very important role in protecting the epithelial cells in the stomach. So there is a layer of a mucus, which is a viscoelastic gel that contains 85% of water and 15% glycoproteins, and this mucus layer impedes hydrochloric acid and pepsin movement from the lumen into the apical cell membrane, and thus protect the cells from hydrochloric acid and the action of pepsin. In addition, epithelial cells secrete bicarbonate at the epithelial surface, so the pH at the epithelial surface is kept at around 7, whereas in the lumen of the stomach the pH is between 1 to 6. In addition, there are cells that secrete prostaglandins, and prostaglandins are important in increasing blood flow to the stomach and help repairing any damage to the epithelial cells.
What are the functions of the stomach? The function of the stomach is first to store, mix, and grind food, so it will transform large particles into smaller particles. It continues digestion that starts in the mouth by the salivary amylase. The presence of HCl in the stomach solubilizes food particles and may disrupt the connective tissue which forms the extracellular matrix of proteins that we ingest. The presence of acid in the stomach is important in protecting us from infections because it kills a lot of microbes that we ingest, and also the stomach regulates the emptying of dissolved food into the small intestine, because as I've said, there is a muscle, a sphincter called the pylorus, which regulates how much food will go from the stomach into the small intestine.
After the stomach, food continues into the small intestine, which have three important parts. The first part is the duodenum, the second is the jejunum, and then the longest part is the ileum. The small intestine is the most important site for digestion and absorption, and this can take place in the lumen or at the surface of the enterocytes, or sometimes inside the enterocytes. Most of what we ingest is usually absorbed and digested in the first 50 to 70% of the small intestine. It is important to know that there are specific parts of the intestine which are responsible for the absorption of specific nutrients and substances. For example, iron is absorbed from the duodenum, whereas bile acids and vitamin B12 are absorbed exclusively from the ileum. The small intestine also can secrete water, bicarbonate, and also can secrete some hormones that we are going to talk about later on. As you have learned from the histology lectures, there are finguli projections in the lumen of the small intestine, which are the villi that play a very important role in digestion and absorption, and each villus is covered by one layer of intestinal cells called enterocytes, and each villus has its own arteriole, venule, and lacteal. Now the enterocytes differentiate from the crypts up to the villus, and it has a turnover rate of 3 to 5 days, So every 3 to 5 days, millions of cells are shed in the lumen and new cells are formed. Also, it is important to know that the enterocyte is a very specialized cell, and if we look at its cytoplasm, it has multiple convolutions called microvilli, which increase the surface area available for absorption.
So if we calculate the surface area available for absorption, and if first we assume that the small intestine is flat, so it's a mucosal cylinder, we can say that he has a surface area of 3,300 centimeter square. If we add to it the fold of Kerckring, then the villi and microvilli, we can see that the surface available for absorption is approximately 2 million centimeter square, which is more or less equivalent to a double tennis court. This is why the small intestine is a very, very efficient organ for digestion and absorption.
Now the last part of the ileum will connect to the colon by the ileocecal valve to reach the first part of the colon, which is the cecum. Then we have the ascending colon, transverse colon, descending colon, sigmoid, and then the last part, which is the rectum, and the rectum end up by the anus.
The colon and the rectum have multiple functions. This is where some absorption of water and ions takes place. This is where bacterial fermentation of non-absorbed nutrient occurs, and the colon plays an important role as storage of waste and indigestible material, and then elimination through the anus.
Now we'll start with the first set of accessory organs, which are the salivary glands. And as you know, we have three types of salivary glands: the submandibular, the parotid, and the sublingual salivary glands.
And these glands secrete daily approximately 800 to 1,500 milliliter of saliva, which is serous, containing an important enzyme called ptyalin or alpha amylase, which is important for the digestion of starch. And also salivary glands secrete mucus. It's very important to keep in mind that the saliva pH is neutral in the range of 6.8 to 7.8.
The secretion of saliva is controlled by both sympathetic and parasympathetic neurons, and both of them will increase the secretion of saliva from salivary glands. The secretion is increased by sight or smell of food. In addition, the presence of food in the mouth stimulates salivary secretions, which is a reflex that is initiated by chemoreceptors that are present in the mouth. For example, if you put a piece of lemon in the mouth, this will elicit salivary secretion. There are also pressure receptors in the mouth and the tongue, which, when activated, will increase secretion of saliva. The increased secretion of saliva is associated with an increase in blood flow to salivary glands that is mediated by parasympathetic system.
Salivary secretions have several functions. They are important for oral hygiene and lubrication, so as if you are always washing your mouth. This is where the initiation of starch occurs, because, as we said, you have the enzyme amylase, which we are going to talk about later on. Salivary secretions dissolves food molecules and facilitates taste. And also it has antibacterial effect, contains proteolytic enzymes like lysozymes, and also contains immunoglobulin A. Salivary secretion also helps clearance and neutralization of reflux gastric material. So if you have some reflux, so some acid will go from the stomach into the esophagus. Then salivary secretions, because it has a neutral pH, it can somehow neutralize the acid which go into the esophagus. And finally, it aids in speech, because if you have dry mouth, it is very difficult to speak, so you have always to have moist tongue.
Now we move to a second accessory organ, which is the pancreas. It is an elongated organ in the retroperitoneum. The pancreas plays a very important role in digestion, and the secretion from the pancreas go into the pancreatic duct and open in the duodenum by the sphincter of Oddi. The basic secretory unit of the pancreas is called acinus, and the acinar cells or pancreatic cells secrete digestive enzymes, whereas the pancreatic duct epithelial cells secrete carbonate. So the pancreatic cells secrete what we call the pancreatic juice, and it contains multiple digestive enzymes important for the digestion of proteins like trypsinogen, chymotrypsinogen, proelastase, procarboxypeptidase A and B. It's important for the digestion of carbohydrates in the presence of pancreatic amylase. Also, it's very important for the digestion of fat, as it secretes pancreatic lipase, colipase, and prophospholipase. And it is important to know that all these enzymes are secreted into the lumen in inactive forms. And when they are in the lumen, the trypsinogen is activated into trypsin by an enzyme present on the enterocyte. It's called enteropeptidase or enterokinase. And so this will transform trypsinogen into trypsin, and then trypsin will transform all the other inactive enzymes into active enzymes. So, for example, chymotrypsinogen will be transformed to chymotrypsin, procarboxypeptidase to carboxypeptidase, etc. In addition, it is important to know that pancreatic cells contain trypsin inhibitor, which inhibit trypsin activation until after they have been secreted into the intestine. So the acinar cells contain all the enzymes responsible for the digestion of carbohydrates, proteins, and lipids. So the question now is, how does the pancreas protect itself from autodigestion? So there are many mechanisms. First, the enzymes are not free in the cytoplasm, but they are packaged in membrane-bound zymogen granules in the acinar cells, so these enzymes cannot digest the cells themselves. Second, the pH in the zymogen granules is acidic, so this acid will inactivate any trypsin activation. Third, there is this trypsin inhibitor in the cytoplasm of acinar cells that can also inhibit trypsin if it is activated. Fourth, most of the enzymes are secreted as inactive precursors, and they are activated by the presence of enteropeptidase in the lumen and not in the pancreas.
The duct cells secrete bicarbonates, and this concentration can rise to 145 milliequivalent per liter, which is 5 times that of plasma, and usually it is stimulated by the hormone secretin, which is secreted by duodenal mucosa when acid food enters the duodenum. So when acid enters the duodenum, the pH is low, and when the pH is low, then there is activation of secretin, and parasympathetic innervation, which increases the secretion of bicarbonate from the pancreas, and this is important for two reasons. First, peptic activity of gastric juices is blocked, and second, we need the appropriate pH for the pancreatic enzymes to work. So pancreatic enzymes work in neutral pH, whereas, as we have mentioned before, pepsin in the stomach works in acidic medium and is inactivated in neutral medium. If we want to know what stimulates the pancreas to secrete digestive enzymes and what stimulate the duct to secrete bicarbonate, we have to look at the receptors that I have, and we can see here that the acinar cells contain receptors for cholecystokinin, acetylcholine, and gastrin releasing peptide. So if we have increase in CCK production or increase in parasympathetic input to the pancreas, there will be an increase in pancreatic secretion. Similarly, there are receptors for acetylcholine and secretin on the duct cells. So if we have increased secretin or increased parasympathetic activity, we'll have increased bicarbonate secretion from the pancreas. Now, the other important accessory organ is the liver, which will be covered in detail by Dr. Barada, but just I will mention a few things. The liver cells or hepatocytes are important cells in the liver that secrete bile and other components, and these secretions go into bile canalicula or ducts, and then these ducts form what we call left hepatic and right hepatic ducts. These two ducts fuse to form the common hepatic duct, then they join the cystic duct from the gallbladder to form the common bile duct, and the common bile duct opens into the duodenum by an important sphincter called sphincter of Oddi. The liver secretes bile, and bile contains 6 major ingredients, which are the bile salts, lecithin, which is a phospholipid. Bile contains bicarbonate ions and other salts. They contain cholesterol, bile pigment, and metabolic end products, and trace metals. Bile salts and lecithin are synthesized in the liver. They help solubilize fat in the small intestine. Bicarbonate ions are secreted by epithelial cells lining the ducts, and they are important to neutralize the acid in the duodenum and have the similar function to that of bicarbonate secreted by the pancreas. Now, the other ingredients represent substances extracted from blood and excreted via the bile. Here on this slide, this is just a summary of all the different organs of the GI tract and their function, which you can just review. Now, how does our gastrointestinal system digest and absorb nutrients that we ingest? First, we'll start with carbohydrates, and as you know, carbohydrates are present in bread, rice, pasta, cornflakes, milk, fruits, lollipops, and all sweeties. We ingest daily approximately 250 to 300 grams of carbohydrates, and this constitutes 45% of our total energy requirements. And approximately 2/3 of what we ingest as carbohydrates is in the form of starch, it's form of amylose or amylopectin, and the rest is in the form of disaccharides like sucrose, which is table sugar, or lactose, which is milk sugar, and also we ingest some undigestible carbohydrates like cellulose, hemicellulose, and pectin, which are called fibers. The digestion of carbohydrates starts in the lumen, and this is an example of starch composed of a long chain of glucose moieties at alpha-1,4 linkages and branches at alpha-1,6 linkages. Now amylase, which is secreted by the salivary glands and pancreas, attack the alpha-1,4 linkages at the last 2 or 3rd moiety at the end of the chain, but cannot attack alpha-1,6 linkages. This is why with the digestion of amylase we will get maltose, maltotriose, and alpha-limit dextrins, which are small chains containing alpha-1,4 linkages and alpha-1,6 linkages of glucose moieties. Then the digestion continues at the brush border membrane. So we have maltase, which can transform maltose into glucose. We have sucrase isomaltase, which transform alpha limit dextrins into glucose, and then glucose is absorbed. Lactose is transformed by an enzyme called lactase to galactose and glucose. Sucrose is transformed by sucrase to glucose and fructose, and then they are absorbed. And the only way by which the enterocyte can absorb carbohydrate is in the form of monosaccharides. So our enterocyte cannot absorb disaccharide or trisaccharides. So everything has to be broken down to monosaccharides to be absorbed. How are monosaccharides absorbed? Well, they are absorbed by 2 important transporters. The first one is called sodium glucose link transporter 1, which takes 1 glucose or galactose moiety with sodium. The other transporter is called GLUT5, which transports fructose into the cell, and then after being in the cell they are moved to the systemic circulation by another transporter called GLUT2. Now, as we have mentioned, lactose is a major carbohydrate in milk, and it is digested by lactase to glucose and galactose. And usually lactase is present at birth, and we are genetically determined to lose this lactase during childhood, especially in Asians and Africans, and this is less prevalent in the West. This is why we have some intolerance to lactose, because the undigested lactose will reach the colon. It is digested by bacteria with production of gas and short-chain fatty acids, leading to diarrhea. And the treatment of that is by lactase enzymes, which can be present as pills, and we can take them when we drink milk, or most people just avoid milk, and these symptoms are less with milk products, because like cheese or labne, because they have lower levels of lactose. Now we move to proteins, and proteins are found in poultry, fish, hamburger, meat, etc. Our daily requirements is approximately 40 to 50 grams. However, the American diet contains much more, 70 to 90 grams. This constitutes 10 to 15% of our daily caloric intake, and proteins that we ingest is broken down to peptide fragments in the stomach first by pepsin, and then it is digested by the pancreatic enzymes trypsin, chymotrypsin, elastase, which are called endopeptidases, and carboxypeptidase A and B, which are called exopeptidases. peptidases. For example, this is a peptide chain which is broken down by endopeptidases into two smaller peptides, and then in the presence of carboxypeptidases A and B, which are exopeptidases, these will be broken down into amino acids and smaller peptides. This intraluminal hydrolysis results in approximately 40% of free amino acids and 60% of oligopeptides. These oligopeptides are further digested by peptidases which are present on the brush border membrane, and these are endopeptidases, aminopeptidases, carboxypeptidases, and dipeptidases. And also there are some peptidases which are present in the cytoplasm of epithelial cells, which can break down these peptides into smaller peptides or amino acids which can be absorbed. The amino acids are taken up by the enterocyte by specific transporters which are linked to sodium, and the dipeptides and tripeptides are taken up by the cell by transporters linked to hydrogen. And there are many different transporters, and there are more than 20 different transporters which have been identified, and each one transports a specific group of amino acids. Now, fat is present in butter, cheese, chocolate, cake, meat, oil, etc. And just important to keep in mind that there is nothing called fat-free meat. There is always some fat in the meat that we ingest. And the American diet contains approximately 70 to 100 grams of fat. It constitutes 1/3 of daily caloric intake, and most of the fat we ingest is in the form of long-chain triglycerides, and we ingest some cholesterol. The major digestive enzyme for triglycerides is lipase, which is secreted by the pancreas. So triglycerides in the presence of lipase will give us monoglycerides plus 2 fatty acids. This seems very simple, however it is much more complicated, and the problem is that triglycerides is not soluble in water, so fat will form large lipid droplets in the stomach, and lipase can attack triglycerides only on the surface of this droplet. So we have a large volume to surface ratio, so it will take a very long time for lipase to digest lipid. So these large droplets have to be divided into small droplets to be digested by lipase, and therefore we are increasing the surface over volume ratio. Imagine, for example, you have a vinegar and oil in a bottle, and then you shake the bottle. You can see that oil will form small droplets. However, if you leave the bottle to rest, and then you can see that oil will form large droplet on the surface of the vinegar. And this is the same what happens in our stomach and small intestine. So to be able to divide these large droplets into smaller droplets, we have the mechanism called emulsification. And emulsification has two components. First, mechanical disruption of these droplets into small droplets by contractile activity in the stomach and small intestine. This is similar when you shake oil and vinegar. And then we need emulsifying agents that prevent reaggregation into large droplets. And these agents are phospholipids, which are present in food, and phospholipids and bile salts, which are secreted in bile. So they are secreted by the liver. This is an example of an emulsifier, which is bile salt, and this is called an amphipathic molecule because it contains a polar side, which is formed by the hydroxyl groups and carboxyl groups, and a non-polar side, which is formed by the methyl groups. So if you look at the tertiary structure of bile salts, there is one side which is non-polar and another side which is polar. So after mechanical disruption of these fat globules into smaller ones, the bile salts and phospholipid will attach to these small droplets from their non-polar side, and then the polar side is from outside, and so by the presence of two polar sides they will repel each other and prevent reaggregation into fat globule, into large fat globule, and this is what you call emulsion droplet. So now we have a large surface area for pancreatic lipase to digest triglycerides into monoglycerides and fatty acids. And now we have another problem, that monoglycerides and fatty acids are not very soluble in water, and they are not directly taken up by the cell, so they will, if they are not preserved, they will be lost in stools, and this is where another function of bile salt come into the picture, with the formation of micelles, which are similar to emulsion droplets but very small, in smaller size, 4 to 7 nanometer in diameter. And here the polar sides of the micelles is from outside and the non-polar from inside. So there is very small amount of monoglycerides and fatty acids which are soluble, which will diffuse into the cells, whereas the other amount of monoglycerides and the monoglycerides and fatty acids are in the micelle. So these micelle will be disintegrated with small amount of monoglycerides and fatty acids in solution that will be absorbed, and this micelle is reformed again, and so there is a continuous disintegration and formation of these micelles. Just keep in mind that the micelle is not absorbed into the small intestine, but monoglycerides and fatty acids are absorbed. Now, while in the cell, the monoglycerides and fatty acids are reformed into triglycerides inside the enterocyte, and then they leave the cell into the as chylomicron into the lacteal. So they go into the lymphatic system and not the systemic circulation. So this is for triglyceride. Now for cholesterol, cholesterol requires only solubilization by bile salts, so we don't need lipase for the digestion of cholesterol. So we have formation of mixed micelles, then they will diffuse into the intestinal cells, and then they are absorbed also into the lymphatic system. ZF. Now there is another type of triglycerides, which is the medium chain triglycerides that require only hydrolysis, so the bile salts are not needed, and the characteristic of these triglycerides are that they are soluble in water. So triglycerides with medium chain fatty acids are hydrolyzed by lipase with the formation of medium chain fatty acids, and these can diffuse directly into the intestinal cells, and they are absorbed into the circulation, directly into portal circulation and not into lymphatics. This type of triglycerides is found in small amount in the in nature, so they are found in coconut oil, but usually they are manufactured and they are used in patients who have some part of their intestine resected, so they cannot absorb long chain triglycerides easily, and this is what you call short bowel syndrome. To these patients we give medium chain triglycerides, which is easily absorbed. We also ingest vitamins, and the fat-soluble vitamins, which are vitamin A, D, E, and K, are absorbed with fat, and so they are solubilized in my cells, then absorbed. The water-soluble vitamins are absorbed by diffusion or mediated transport, except vitamin B12. Vitamin B12 binds to a protein called R binder, which is secreted by salivary glands and by the stomach, and these B12 R binder are hydrolyzed by protease secreted by the pancreas, and this happens in the duodenum. Then B12 will bind to intrinsic factor, which is secreted by parietal cells in the stomach, and then B12 intrinsic factor will go to the ileum, they bind to a receptor in the ileum, and then B12 is absorbed. Iron, 10% of ingested iron is absorbed. Each day it is actively transported into epithelial cells in the duodenum and incorporated into a protein called ferritin, and this is how it is stored. The excess iron is released on the blood side and circulates bound to transferrin. It's important to know that absorbed iron cannot be excreted, so it's not excreted in urine, it's not excreted in stools. Whenever it is absorbed, it has to be used, or if you have excess iron, it may be deposited in the heart, in adrenals, in the kidneys, in the liver, and may cause problems. This is why it is very important for our body to control iron absorption, and usually absorption depends upon body iron store and type of food that we ingest. So what will happen if you have iron deficiency, for example? Then the body will tell the duodenal cells to decrease formation of ferritin. If the ferritin in the cells is low, then more iron is absorbed. If we have excess iron in our circulation, then our body will send messages to the duodenal cells to increase formation of ferritin. Whenever you have a large amount of ferritin, so larger amount of iron will bind to ferritin, and smaller amount will be absorbed. And then our body gets rid of iron by the shedding of the intestinal cells, and as I've mentioned previously, millions of cells are shed every few days and new cells are formed, and this is how the iron is excreted in the stools. In general, iron from meat is better absorbed from non-heme origin, so from vegetable origin. It is better absorbed in ferrous form than in ferric form, and there are many things that inhibit iron absorption. For example, phytate, which is present in some vegetables. Egg decrease iron absorption because of the presence of phosphatin. Calcium, oxalate, all these inhibit iron absorption, whereas vitamin C enhances iron absorption. Now, in addition to food that we ingest and vitamins, there is a large amount of water or fluid which is secreted into the intestine and has to be reabsorbed. And every day approximately 9 liters enter our intestine, and most of this will be absorbed. So in 24 hours, more than 8 liters of fluid are absorbed by the small intestine and the colon. So if you consider that the salivary glands secrete approximately 1.5 liters of saliva every day, the stomach secretes 2 liters of hydrochloric acid, the liver secretes half a liter, the pancreas 1.5 liters, and the small intestinal cells secrete approximately 1.5 liters of secretion. Then all this, approximately 6.7 liters, are absorbed every day from the small intestine, and approximately 1.5 liters from the colon. And the feces usually contains only 100 milliliter of water. So it's just to show you how efficient our small intestine in absorbing the food and water. Now at the cellular level, how is water absorbed? So water absorption depends on sodium absorption, and there are two ways how sodium is absorbed. All depends on the presence of a pump called sodium potassium ATPase pump, and this pump will pump sodium outside of the enterocyte into the interstitial medium, and then water will follow. Now sodium is taken up in the cell down its electrochemical gradient through two important transporters. The first one is sodium hydrogen transporter, and so this will take sodium inside the cell and secrete H+ into the lumen. And the second one is the glucose transporter, which I have talked about previously, which is called sodium glucose link transporter, and so this absorbs sodium with glucose at the same time. So now we have increased sodium in the interstitial area, and water has to follow. So water is not absorbed through the cell but between the cells, and then it will enter the interstitium and goes into the circulation. But our enterocytes are also capable of secreting water, and this is because of the presence of a chloride channel called the cystic fibrosis transmembrane regulator, or CFTR, and these channels are usually opened when you have increased cyclic AMP. So chloride will be secreted into the lumen, and then water will follow. These chloride channels are open with some toxins like cholera toxin, heat stable enterotoxins, heat labile enterotoxins, and this is when you get diarrhea, and this is secondary to the opening of these chloride channels. Now, how are gastrointestinal processes regulated? So how to regulate the secretion from the parietal cells? How do we control secretion from the pancreas and liver? And how do we control absorption from the intestines? Well, it's important to know that the absorption of nutrients is not related to the nutritional status of the individual. So an obese patient or a non-obese patient will not have different rate of absorption. When we talk about regulation, we have to talk about reflexes, and in any reflex we need a stimulus that stimulates a receptor, and this receptor will trigger a reflex that influence an effector. So in the GI tract, the stimuli are sight, smell or taste of food, distension of the lumen, chyme osmolarity and acidity, and concentration of digestion products, and these will stimulate receptors which are located in the wall of the tract, and these are mechanoreceptors, osmoreceptors or chemoreceptors, and this will trigger reflexes that influence the effectors, and the effectors in the GI tract are the muscles in the wall, so they affect motility and contraction, and exocrine glands which are present in the pancreas, or can also affect some cells scattered in the stomach and duodenum, like parietal cells, G cells, etc. There are two important ways to regulate GI processes. We have the neural regulation and hormonal regulations, and in most instances we have both neural and hormonal regulation. We start by the neural regulation of GI processes, and these are regulated by the enteric nervous system and the central nervous system. So the enteric nervous system, which is formed by the myenteric and submucosal plexuses, and the neurons in these plexuses synapse with other neurons or end on smooth muscles, glands, or epithelial cells. And also the neurons in one plexus can synapse with neurons in a different plexus. One characteristic of these cells or these nerves is that stimulation at one point can be conducted both up and down the tract, and in general the myenteric plexus influences smooth muscle activity, so it affects contractions and motility, and the submucosal plexus influences secretory activity. Now also the regulation of GI processes is controlled by the central nervous system, so the nerve fibers from autonomic nervous system enter and synapse with nerves in both plexuses or can affect directly the effector cells. Now we have two types of pathways, the long and the short neural reflex pathways. First we start by the short reflexes. So if we have a stimulus in the lumen, this will stimulate the receptors, and then this will affect the effector cells, smooth muscle or gland, and then we will have a response, and this is what we call short reflex. Or the stimulus will affect a receptor, then a message is sent to the central nervous system, and then the central nervous system will affect the nerve plexuses or will work directly on the effector cells to get a response. Now, other than neural regulation, we have hormonal regulation of GI processes, and these are hormones which are secreted by endocrine cells which are scattered throughout the epithelium of the stomach and the small intestine. One surface of each endocrine cell is exposed to the lumen of the GI tract, and so substances in the chyme stimulate the cell to release its hormones into the blood, which is called endocrine type of hormones, or into adjacent cell, which is called a paracrine form. So if you have a sensor cell here and there is a stimulus, this cell will secrete a hormone which go into the circulation and affect a target cell, for example cholecystokinin, and we are going to talk about this later on. Or it can affect by paracrine mediators, so for example histamine. If you have a stimulus of the enterochromaffin like cell, these cells will secrete histamine, and histamine will have an effect on an adjacent cell, and this is what we call paracrine way. There are many different hormones which are important in the GI regulation, and I'm going to discuss only 5 of those, which are gastrin, cholecystokinin, secretin, glucose isotropic peptide, and somatostatin. All of these are peptides, and we'll start by the first one, which is gastrin. And gastrin is produced by G cells in the antrum. The stimuli for its production is the presence of peptides, amino acids, and neural stimulation. What inhibits gastrin secretion is acid and somatostatin. What's the function of gastrin? Well, the most important function of gastrin is to stimulate acid secretion, and also stimulates gastric motility, so increase gastric contractions, and also increase small intestinal and large intestinal motility. The second hormone is cholecystokinin, and cholecystokinin is produced by eye cells in the small intestine. The stimuli for its secretion is the presence of amino acids and fatty acids in the lumen. The most important function of cholecystokinin is to stimulate pancreatic enzyme secretions, and also to a lesser extent it stimulates bicarbonate secretion from the pancreas. In addition, it stimulates secretion of bicarbonates from the liver and also contracts the gallbladder and relaxes the sphincter of Oddi. In addition, cholecystokinin can inhibit acid secretion and inhibit gastric motility. The third hormone is secretin, and secretin is secreted by S cells present in the duodenum. The stimuli for its secretion is acid, and the most important function of secretin is the secretion of bicarbonates from the pancreas, and to a lesser extent stimulates enzyme secretion from the pancreas, and it also increases bicarbonate secretion from the liver. In addition, it inhibits acid secretion and inhibits gastric motility like cholecystokinin. The fourth one is glucose-dependent isotropic peptide. It is produced by the small intestine. The stimuli for its secretion is the presence of glucose and fat, and what it does is stimulates insulin secretion from the pancreas. The last one is somatostatin, which is present in the D cells in the antrum and in the pancreas. The stimulus for its secretion is the presence of acid in the stomach, and somatostatin is an inhibitory hormone that inhibits everything. So it inhibits acid secretion, it inhibits pancreatic enzyme secretion as well as insulin secretion, it relaxes the gallbladder and inhibits small intestinal and large intestinal motility. Now there are some characteristics of GI hormones that we need to know. Each hormone participates in a feedback control system that regulates some aspect of the GI luminal environment, and each hormone may affect more than one type of target cell. For example, cholecystokinin, in the presence of fatty acids and amino acids in the small intestine, triggers CCK secretion from cells in the small intestine into the blood. The circulating cholecystokinin stimulates secretion of digestive enzymes by the pancreas. At the same time, it causes the gallbladder to contract, delivering bile salts to intestines for micelle formation, and as fatty acid and amino acid are absorbed, CCK release is stopped. Another characteristic of these hormones is the potentiation. So a single effector cell may contain receptors for more than one hormone, paracrine agents and neurotransmitters, so a variety of inputs can affect the cell's response. One example of this is interaction between CCK and secretin. As you can see here, this is the effect of secretin and CCK on pancreatic bicarbonate secretion. As you can see, secretin is more potent than CCK in increasing bicarbonate secretion. However, if you put them together, their combined effect is more than the mere addition of the effect of CCK and secretin alone. So this is what we call potentiation. So CCK potentiates the effect of secretin in increasing pancreatic bicarbonate secretion. This is why sometimes small changes in the plasma concentration of one GI hormone can have large effects on the actions of another GI hormone. Now there are other hormones which you need to know about, and these are leptin and ghrelin. And leptin is released from fat cells and influences food intake, so induces satiety and has an influence on metabolic rate. Ghrelin is released from the stomach during fasting and stimulates hunger. Both hormones act on receptors in the hypothalamus, and leptin was found to be important in obesity, and it has been found that obesity is associated with a decreased sensitivity to leptin, and therefore there is an increase in serum leptin. The neural and hormonal control of the gastrointestinal system is in large part divisible into three phases: cephalic, gastric, and intestinal, according to where the stimulus is perceived. This is an example of a cephalic phase where you just see the food, you smell it, and or when you are in a restaurant and you have the mesa in front of you and you are waiting for other people to arrive, this is what will happen. So this is initiated when receptors in the head are stimulated by sight, smell, taste, and even chewing. Various emotional states can also initiate this phase. The afferent pathways for these reflexes are primarily mediated by parasympathetic fibers carried in the vagus nerve. These fibers activate neurons in the gastrointestinal nerve plexuses, which in turn affect secretory and contractile activity. So this is an example. This is a cephalic phase. So you have activation of the dorsal vagal complex, which will send afferent to the stomach to secrete hydrochloric acid, secrete pepsinogen, secrete restrict factor, secrete gastrin. Also there are some afferent which go to the pancreas to secrete pancreatic enzymes, to the gallbladder to contract, and to the sphincter of Oddi to relax. The second phase is the gastric phase, and this is initiated by 4 types of stimuli in the stomach, which is distension of the stomach, the presence of acidity, the presence of amino acids and peptides, and this is mediated by short and long neural reflexes and by the release of the hormone gastrin. So as you can see here, with the distension of the stomach, there are some afferent neural input into the dorsal vagal complex, which will send efferent fibers to the different parts of the GI tract. In addition, we have short neural reflexes in the stomach itself to increase secretion of gastrin and hydrochloric acid and pepsin, and also gastrin itself will increase H+ secretion and pepsinogen secretion. The third phase is the intestinal phase, and this is when the stimulus is in the small intestine, and this is initiated by distension, the presence of acidity, the osmolarity of the content, and some digestive products, and this is mediated by both short and long neural reflexes and by the GI hormones secretin, cholecystokinin, and glucose dependent isotropic peptide. Now all these phases do not occur in temporal sequence except at the very beginning of a meal. However, during ingestion, digestion, and absorptive periods, reflexes characteristic of all 3 phases may occur simultaneously. Keeping in mind the neural and hormonal mechanisms available for regulating gastrointestinal activity, we can now examine the specific contractile and secretory processes that occur in each segment of the gastrointestinal system. And we are going to follow the food from the time we chew to the time we defecate. And we start with the chewing, and chewing is controlled by the somatic nerves to the skeletal muscles of the mouth and jaw. In addition to the voluntary control of these muscles, rhythmical chewing motions are reflexly activated by the pressure of food against the gums, hard palate, at the roof of the mouth and tongue. Activation of these mechanoreceptors leads to reflexive inhibition of the muscles holding the jaw closed. So the resulting relaxation of the jaw reduces the pressure on the various mechanoreceptors, leading to a new cycle of contraction and relaxation. Now swallowing. Swallowing is a complex reflex which is initiated when pressure receptors in the walls of the pharynx are stimulated by food or drink forced into the rear of the mouth by the tongue. These receptors send afferent impulses to the swallowing center in the brainstem medulla oblongata, and this center then elicits swallowing via efferent fibers to the muscles in the pharynx and esophagus, as well as to the respiratory muscles. As the ingested material moves into the pharynx, the soft palate elevates and lodges against the back wall of the pharynx, preventing food from entering the nasal cavity. Impulses from the swallowing center inhibit respiration, raise the larynx, and close the glottis, and the glottis is the area around the vocal cords and the space between them, thus keeping food from moving into the trachea. As the tongue forces the food farther back into the pharynx, the food tilts a flap of tissue called the epiglottis backward to cover the closed glottis, thereby preventing food from entering the trachea. And this is what we call to prevent aspiration of food. Now, the esophageal phase of swallowing begins with relaxation of the upper esophageal sphincter, and immediately after the food has passed, the sphincter closes, the glottis opens, and the breathing resumes. Once in the esophagus, the food moves toward the stomach by a progressive wave of muscle contractions that proceeds along the esophagus, compressing the lumen and forcing the food ahead. Such waves of contraction in the muscle layers surrounding a tube are known as peristaltic waves. One esophageal peristaltic wave takes about 9 seconds to reach the stomach. We can measure the pressure in different parts of the esophagus, and as you can see, when you swallow, there is relaxation of the upper esophageal sphincter, then a peristaltic wave will spread from the upper esophagus to the lower esophagus, and you can see that upon swallowing directly, when you swallow, there is relaxation of the lower esophageal sphincter, and this relaxation continues during the whole period of swallowing, and you can see it better here. So when you swallow, there is relaxation of the lower esophageal sphincter, allowing food to go into the stomach, and at the same time we have some relaxation of the stomach to accommodate food that it receives. Now, what happens to the hydrochloric secretion from the stomach during cephalic and gastric phases? And I will here remind you that hydrochloric acid is secreted by parietal cells present in the stomach, and parietal cells have 3 stimulatory receptors, which are a receptor for gastrin, a receptor for acetylcholine, and a receptor for histamine. So during the cephalic phase, we have stimulation of the central nervous system that will send impulses to the enteric nervous system, as well as directly the G cells, histamine cells, and parietal cells. And so if you have stimulation of gastrin, then we'll have increased gastrin secretion, which will lead to direct activation of parietal cells, and will lead increase of histamine from enterochromaffin-like cells, and all these will increase parietal cell secretion of hydrochloric acid. During the gastric phase, when we have luminal distension or the presence of amino acids and peptides, this will stimulate gastrin secretion. So gastrin secretion will increase, leading to increase parietal cell secretion. In addition, as we have said, in the gastric phase, we have also the stimulation of the short neural reflexes and long neural reflexes, which will further increase acid secretion. Now, how to regulate acid secretion? So if acid secretion is too high, it will reach the antrum, and in the antrum we have stimulation of D cells that will secrete somatostatin, In addition, HCl itself will inhibit gastrin secretion from G cells. I will repeat. So if acid secretion reaching the antrum is high, then H plus will directly inhibit secretion from the G cells. In addition, there is stimulation of D cells which secrete somatostatin, and somatostatin is an inhibitory hormone that inhibit secretion of HCl from G cells, and this is how HCl secretion is regulated. In addition, the stomach secretes pepsinogen from chief cells, and this is due to the stimulation by the nervous system, and secretion of pepsinogen parallels the secretion of HCl from the stomach. And pepsinogen, with the action of H plus, will be transformed into pepsin, and pepsin further transforms pepsinogen into pepsin, which is important for protein digestion. The pepsinogen is present in inactive precursors, and I will remind you again that pepsin is only active in the presence of high hydrochloric acid, and it becomes inactivated in the small intestine. And the presence of pepsin accounts for 20% of total protein digestion. Now, how about gastric motility? An empty stomach has a volume of only 50 milliliter. However, it will relax when food reaches the stomach, and this is what we call receptive relaxation, which will increase the volume of the stomach to 1.5 liters. And this receptive relaxation is mediated by the parasympathetic nerves, with coordination by the swallowing center in the brain. Both nitric oxide and serotonin are released by the enteric nervous system, and these will mediate this relaxation. In addition, when food reaches the stomach produces peristaltic waves in response to the arriving food. Each wave begins in the body of the stomach, and it produces only a ripple as it proceeds towards the antrum, a contraction too weak to produce much mixing of the luminal contents with acid and pepsin. As the wave approaches the larger mass of whole muscle surrounding the antrum, however, it produces a more powerful contraction which both mixes the luminal content and closes the pyloric sphincter. The pyloric sphincter muscles contract upon arrival of a peristaltic wave. As a consequence of the sphincter closing, only a small amount of chyme is expelled into the duodenum with each wave, and most of the enteral contents are forced backwards toward the body of the stomach, thereby contributing to the mixing activity in the antrum. And these peristaltic waves continue mixing of the contents of the stomach with pepsin and acid. Here I will remind you again that the lower esophageal sphincter prevents the retrograde movement of the stomach contents from entering the esophagus. Now, what is responsible for producing gastric peristaltic waves? The rhythm, which is 3 per minute, is generated by pacemaker cells in the longitudinal smooth muscle layer. These smooth muscle layers undergo spontaneous depolarization-repolarization cycles known as the basic electrical rhythm of the stomach. These slow waves are conducted through gap junctions along the stomach's longitudinal muscle layer, and also induce similar slow waves in the overlying circular muscle layer. In the absence of neural or hormonal input, however, these depolarizations are too small to cause significant contractions. Excitatory neurotransmitters and hormones act upon the smooth muscle to further depolarize the membrane, thereby bringing it closer to the threshold. Action potentials may be generated and the peak of the slow wave cycle if threshold is reached, and may thus cause larger contractions. The number of spikes fired with each wave determines the strength of the muscle contractions. Thus, whereas the frequency of contraction is determined by the intrinsic basic electrical rhythm and remains essentially constant, which is 3 per minute, the force of contraction and therefore the amount of gastric emptying per contraction are determined reflexly by neural and hormonal input, the enteral smooth muscle. The initiation of these reflexes depends upon the content of both the stomach and small intestine. All the factors previously discussed that regulate acid secretion can also alter gastric motility. For example, gastrin in sufficiently high concentrations increase the force of enteral smooth muscle contractions. Distension of the stomach also increases the force of enteral contractions through long and short neural reflexes triggered by mechanoreceptors in the stomach wall. Therefore, after a large meal, the force of initial stomach contractions is greater, which results in a greater emptying per contraction. However, if you have distension of the duodenum, presence of fat, high acidity, hypertonic solutions, these will decrease the force of contraction. So this is how our small intestine will tell the stomach that I have too much to digest and absorb, so stop sending me material to digest, and then the stomach will decrease the force of contraction and will send less food to the duodenum to be digested. Now, the content of the stomach plays also an important role in gastric emptying. So if we have a liquid meal, this will be emptied quicker than semi-solid meal or solid meal. The rate of gastric emptying is also related to caloric content of the meal. So if we have NaCl, this will be emptied quickly. However, the more caloric intake of a glucose solution, the slower is the emptying of the stomach. So glucose with a 0.2 kilocalorie per ml will be emptied much quicker than a glucose solution containing 1 kilocalorie per ml. Also, the tonicity of the solution is important. So isotonic solutions are emptied faster than hypotonic solutions, which are emptied faster than hypertonic solutions. Now, as I said, we have always 3 contractions per minute, and with every contraction a small amount of material will go into the duodenum. So this is what we call pulsatile flow of food into the duodenum, and this will help the duodenum having enough time to digest and absorb food. So what goes into the stomach is bolus, large particles containing triglycerides, protein, starch, monosaccharides, disaccharides, salivary secretions, water and ions. And what leaves the stomach are small particles, less than 2 millimeters, emulsion, triglycerides with small amounts of 2-monoglycerides and free fatty acids. What leaves the stomach is protein with small amount of peptides and amino acids. We have starch and 20% oligosaccharides, and also what leaves the stomach is monosaccharides, disaccharides, water, ions, and the pH of what leaves the stomach is acidic, so it has low pH. So now we are in the intestinal phase, as the chyme is in the small intestine. And as we have previously mentioned, the stimuli in the intestinal phase is increased acid, increased fat, increased hypertonic solutions, and distension of the small intestine, and this will stimulate neural receptors and also stimulate secretion of enterogastrons, which are CCK and secretin. So with stimulation of neural receptors through long neural reflex and short neural reflexes, we get decrease in acid secretion and decrease in gastric emptying, and also with increased enterogastrons we have also decrease in acid secretion and decrease gastric emptying. And this is the way how the small intestine will tell the stomach that I have too much acid and I have too much food, so stop sending me more. This increase in enterogastrons will also affect the pancreas. So in the presence of fatty acids and amino acids in the small intestine, we have increased CCK secretion and therefore increase in plasma CCK, which will stimulate the pancreas to increase enzyme secretion, and therefore there is increased flow of enzymes into small intestine with digestion of fats and proteins. And at the same time, with the presence of acid in the duodenum, we have increase in secretin secretion. Secretin will increase bicarbonate secretion from the pancreas and will help in neutralization of intestinal acid, so that it will create the neutral environment for the pancreatic enzymes to work. Now CCK also has an effect on the gallbladder, so it will increase gallbladder contraction and it will relax the sphincter of Oddi, resulting in increased bile flow to the duodenum. So now bile in the duodenum will be used for fat digestion and absorption, and then it will go to the ileum where it is absorbed from the ileum, and then it will circulate into the blood. It will be taken up by the liver and secreted again into the duodenum. And during digestion, all the bile pool will be circulating many times, and this is what we call enterohepatic circulation of bile salts. Now, when everything is digested and absorbed, the sphincter of Oddi will close and the gallbladder will be relaxed. During the interdigestive period, there is always some secretion of bile from the liver. This bile will reach the ampulla, which is contracted, so the bile will be shifted back to the gallbladder where it is concentrated, and then when we ingest food, then the gallbladder will contract and all its contents will go into the duodenum. Now, pancreatic enzymes in the duodenum will digest fat, carbohydrates, proteins, and nucleic acids to fatty acids, sugars, amino acids, and nucleotides. So trypsin, chymotrypsin, elastase will break peptide bonds to form peptide fragments, and then carboxypeptidase splits off terminal amino acids from carboxyl end of peptides. Ribonuclease and deoxyribonuclease split nucleic acids into free mononucleic acids. Amylase will split polysaccharides into maltose, maltotriose, and alpha-linolextrins, and lipase splits off two fatty acids from triglyceride, as we have previously mentioned in the digestion of different nutrients. Now we move to motility of the small intestine, and in contrast to the peristaltic waves that sweep over the stomach, the most common motion in the small intestine during digestion of a meal is a stationary contraction and relaxation of intestinal segments with little apparent net movement toward the large intestine. So each contracting segment is only a few centimeters long, and the contraction lasts a few seconds. The chyme of the lumen of a contracting segment is forced both up and down the intestine, and this rhythmical contraction and relaxation of the intestine, known as segmentation, produces a continuous division and subdivision of the intestinal contents, thoroughly mixing the chyme in the lumen and bringing it into contact with intestinal wall. These segmenting movements are initiated by electrical activity generated by pacemaker cells in or associated with the circular smooth muscle layer. As with the slow waves in the stomach, this intestinal basic electrical rhythm produces oscillations in the smooth muscle membrane potential that, if threshold is reached, trigger action potentials that increase muscle contraction. The frequency of segmentation is set by the frequency of the intestinal basic electrical rhythm, but unlike the stomach, which normally has a single rhythm, which is 3 per minute, the intestinal rhythm varies along the length of the intestine, each successive region having a slightly lower frequency than the one above. For example, segmentation in the duodenum occurs at a frequency of about 12 contractions per minute, whereas in the last portion of the ileum, the rate is only 9 contractions per minute. Segmentation produces therefore a slow migration of the intestinal contents toward the large intestine because more chyme is forced downwards on the average than upward. The intensity of segmentation can be altered by hormones, the enteric nervous system, and autonomic nerves. Parasympathetic activity increases the force of contractions, whereas sympathetic stimulation decreases it. Now, after most of the meal has been absorbed, the segmenting contraction cease and are replaced by a pattern of peristaltic activity known as the migrating myoelectric complex, or MMC. Beginning in the lower portion of the stomach, repeated waves of peristaltic activity travel about 2 feet along the small intestine and then die out. The next MMC starts slightly farther down the small intestine, so that peristaltic activity slowly migrates down the small intestine, taking about 2 hours to reach the large intestine. By the time the MMC reaches the end of the ileum, new waves are beginning in the stomach and the process repeats. Upon the arrival of a meal in the stomach, the MMC rapidly ceases in the intestine and is replaced by segmentation. A rise in the plasma concentration of the intestinal hormone motilin is thought to initiate the MMC. Feeding inhibits the release of motilin. Motilin stimulates MMCs via both the enteric and autonomic nervous system. Now, the contractile activity in various regions of the small intestine can be altered by reflexes initiated at different points along the gastrointestinal tract. For example, segmentation intensity in the ileum increases during periods of gastric emptying, known as the gastroileal reflex. Large distension of the intestine, injury to the intestinal wall, and various bacterial infections in the intestine lead to a complete cessation of motility, which is called the intestinal reflex. Although the large intestine has a greater diameter than the small intestine, its epithelial surface area is far smaller. Since the large intestine is about half as long as the small intestine, its surface is not convoluted and its mucosa lacks villi. The secretions of the large intestine are scanty, lack digestive enzymes, and consist mostly of mucus and fluid containing bicarbonate and potassium ions. The primary function of the large intestine is to store and concentrate fecal material before defecation. Chyme enters the cecum through the ileocecal sphincter or ileocecal valve, and approximately 1,500 ml of chyme enter the large intestine from the small intestine each day. This material is derived largely from the secretions of the lower small intestine, since most of the ingested food is absorbed before reaching the large intestine. The primary absorptive process in the large intestine is the active transport of sodium from lumen to blood with the accompanying osmotic absorption of water. If fecal material remains in the large intestine for a long time, almost all the water is absorbed, leaving behind hard fecal pellets. The colon also absorbs some of the products formed by the bacteria inhabiting this region. For example, undigested polysaccharides like fibers are metabolized to short-chain fatty acids by bacteria in the large intestine and absorbed by passive diffusion. The bicarbonates the large intestine secretes helps to neutralize the increased acidity resulting from the formation of these fatty acids. These bacteria also produce small amounts of vitamins, especially vitamin K, for absorption into the blood. Other bacterial products include gas, which is flatus, which is a mixture of nitrogen and carbon dioxide with small amounts of inflammable gases, hydrogen, methane, and hydrogen sulfide. Certain foods like beans contain large amounts of undigestible carbohydrates which are metabolized by colonic bacteria with production of gas. So if you have a sandwich of falafel, for example, or you have a breakfast containing full hummus, onions, radish, you have all experienced this, you will develop large amounts of gas because of the presence of undigested fibers. And this is normal in all primates because they cannot digest fibers with the production of gas. So gas comes from different origins, so there are multiple mechanisms that account for gas accumulation. First, we ingest air when we swallow our food. Then there is production of CO2 when there is neutralization of H plus and bicarbonate in the small intestine. So acid is coming from the stomach and bicarbonate is secreted by the pancreas and the liver, and so there is neutralization with formation of CO2. And then the third origin of the gas is the formation of gas of flatus from bacterial fermentation in the colon. Now, also in the colon we have a special kind of motility. So contractions of the circular smooth muscle in the large intestine produce a segmentation motion with a rhythm considerably slower than that in the small intestine, which is one every 30 minutes. Because of the slow propulsion of the large intestine contents, material entering the large intestine from the small intestine remains for about 18 to 24 hours, and this provides time for bacteria to grow and multiply. And then 3 to 4 times a day, and generally following a meal, a wave of intense contractions known as mass movement spreads rapidly over the transverse segment of the large intestine toward the rectum. This usually coincides with gastroileal reflux. Now, unlike a peristaltic wave in which the smooth muscle at each point relaxes after the wave of contraction has passed, the smooth muscle of the large intestine remains contracted for some time after a mass movement. Now, the anus, which is the exit from the rectum, is normally closed by the internal anal sphincter, which is composed of smooth muscles, and the external anal sphincter, composed of skeletal muscle under voluntary control. The sudden distension of the walls of the rectum produced by the mass movement of fecal material into it initiates the neurally mediated defecation reflex. This reflex consists of a contraction of the rectum, relaxation of the internal anal sphincter, but contraction of the external anal sphincter initially, and increased peristaltic activity in the sigmoid colon. Eventually, a pressure is reached in the rectum that triggers reflex relaxation of the external anal sphincter, allowing the feces to be expelled. Also, the defecation is helped by relaxation of a muscle called puborectalis muscle, and the puborectalis muscle keeps usually a certain angle between the rectum and the anal canal so that we don't soil ourselves. During defecation, this puborectalis muscle will relax, straightening the angle between the rectum and the anal canal and making the defecation easier. And what a relief. Okay, that's all, and good luck in your exams.